Okay, so when last we left off, we were looking at this cell. It is a cell in G2 of interphase. Inside the intact nucleus, we have double the amount of DNA. We've doubled the number of chromosomes in this cell. It's all jumbled in there in the form of chromatin, and there is a nucleolus present. There are two asters, two centriole pairs, and other than that, it's chilling out, waiting for mitosis. So let's talk more about the M phase, the mitotic phase. 10% of the cell cycle is mitosis, and that 10% is divided into 5 separate phases. Mitosis starts with prophase, then leads into what's called Prometaphase, which goes to metaphase, Anaphase, Telophase. And then during telophase, a process called cytokinesis occurs. Cytokinesis separates one cell into two. It pinches the cytoplasm of the membrane. So kinesis is movement, cyto referring to the cell or the cytoplasm that's pinched inward, and it moves like this. And so you have one cell looks like this, it pinches inward and then goes bloop and separates. And you have two cells. So cytokinesis is the separation of one cell into two. Now, that is not a phase. Cytokinesis is not a phase of mitosis. It is, in fact, more of an event. It occurs during telophase. So cytokinesis causes one cell, one parent cell, to become two daughter cells. So here is a brief view of the pictures we'll be looking at. Believe it or not, these pictures are important because it may just be that on an exam you have to draw these. Yeah, draw these and relate what's happening. That might be happening on an exam. You think maybe? Just a hint, helpful hint. So, prophase. In prophase, your chromatin is condensing into sister chromatids. And those sister chromatids are joined at the center by the centromere, that little button of DNA holding two identical sister chromatids together. And this forms the X-shaped chromosomes. Like I said, joined at the centromere. The nucleolus disappears because, well, all the chromatin that made up the DNA in the nucleus, and that nucleolus being just a highly condensed area where chromatin is super concentrated, that nucleolus disappears because the chromatin that was super concentrated there is getting wrapped up tightly into these little X-shaped chromosomes. At the same time, the mitotic spindle starts forming. So that's microtubules being produced between asters. And the centriole pairs, those asters, are moving towards opposite poles. That is prophase. So these are the condensing chromosomes here. This is my centriole, and these are my identical sister chromatids. So they're very long, not compact yet, because they're still condensing. There's other important things to point out here. One, our nuclear envelope is intact. So even though the nucleolus has disappeared, the nuclear envelope is still present. These are my centriole pairs. They're moving away from each other, and these are the microtubules growing out towards each other, forming that mitotic spindle. This leads us into Prometaphase. In Prometaphase, the nuclear envelope fragments. So, that nuclear envelope that was intact in prophase now breaks down into little chunks. And when it does that, it exposes our now fully condensed chromosomes to the cytoplasm. And what's going to happen is those mitotic spindle fibers, those tubules, are going to invade the nuclear area. Now that it's exposed because the nuclear envelope fragmented, the spindle fibers work their way in and start attaching to the kinetocores on the centromeres of our fully condensed chromosomes. The microtubules from the asters enter the nuclear area. The mitotic spindle fibers enter the nuclear area. The chromosomes are now fully condensed. They are two sister chromatids joined at the centromere, and they have kinetocores present, which is good because those microtubules are now binding to the kinetocores. At the same time, our asters are going to pretty much finish moving towards opposite poles. And as the asters keep moving towards opposite poles and the microtubules attached to the kinetocores, they start pushing those chromosomes back and forth in the cell. So pushing them around, moving them ever so slowly towards the center. So here is Prometaphase. This little white stuff here was our nuclear envelope that is now fragmented. You can see our asters are now at opposite poles, and the spindle fibers have invaded the nuclear area and are attached to our chromosomes, these black dots being the kinetocores, so they're attaching to those kinetocores. So, as prometaphase comes to a close, those chromosomes are bumped and pushed and pulled back and forth around in the cell until they reach the center of the cell. So if you think of the cell as a sphere, like this, the center of it is right here. So if you were to sort of open the two halves of a globe, the center is this part here. And if you're looking down on it, it's sort of a plane or a plate in that sphere. So that is called the metaphase plate. Metaphase is characterized by having the chromosomes lined up on the metaphase plate. So our asters are on opposite poles. The chromosomes align at the exact middle at the metaphase plate, and the metaphase plate is not a physical structure. It is a term for the center of the cell. We call it a plate because, like I said, if you were to open that globe in half and look down on it, that circular area kind of looks like a plate. So it's a metaphase plate, not a physical thing, just a word for the center of the cell, the middle of the cell. The chromosomes have aligned there. The mitotic spindle is fully connected between the asters and the kinetocores. And that is metaphase. It's pretty simple. So this line here represents the metaphase plate. Again, not a physical structure, just a term for the middle. And these are our chromosomes lined up along the middle of the metaphase plate. Asters on opposite sides with kinetocores fully attached to microtubules, just like this. And that's going to bring us to our next phase, anaphase. And the thing that's going to happen in anaphase is that our microtubules in the spindle fiber are going to start shortening. And so they're here, so all my chromosomes are here, and this is the middle, right? And so the spindle is on either side. And what they're going to do is they're going to start shortening and pulling towards opposite ends. And as those spindle fibers shorten and pull towards opposite ends, they're going to put stress on either side of that centromere, and they're going to be yanking on it from both directions. And as the forces increase, as the spindle fibers shorten, that centromere is going to end up splitting in half. It'll be cleaved in twain, like a Scotsman with his great sword slicing something in half. That's when you cleave something in twain. Okay, maybe a little too enthusiastic. The centromere split. So the centromere protein, the centromere is split, and the sister chromatids separate. When the sister chromatids separate, the individual chromatids that made up that sister chromatid structure, so if these are my identical chromatids, And this knuckle here is the centromere, and they're joined there. This is my chromosome. When they split, they do this, and they start moving towards opposite poles. So this is the sister chromatids together here, and when they split, they become daughter chromosomes. This is a daughter chromosome. This is a daughter chromosome. And so those daughter chromosomes are going to be moved towards the pole. The daughters are pulled towards the poles. And anaphase ends with the daughters on the pole. So at this point, the cell is elongated into an oval shape. It goes from a nice sphere to this big elongated oval. And it does that because in the next phase, the final phase, telophase, we're going to need to separate that into two cells. And if it's already in an oval, that's going to make it easier for cytokinesis to occur. But we're getting ahead of ourselves. Let's look at anaphase. So each pole has the original amount of chromosomes, the diploid number. Because remember, when we started, we had doubled the number of chromosomes. That is exactly why a sister chromatid chromosome there is an X shape. It's an X shape because, well, put it this way, a normal human cell has 46 chromosomes. If you were to take all of the DNA in G1 of interphase and force it to condense into chromatids, you'd get 46 chromatids. 46 individual chromatids. But DNA doesn't condense during G1. It condenses during mitosis, which occurs after DNA is doubled in the S phase of interphase. So because we have doubled DNA, instead of just getting a single chromatid when we condense, we get sister chromatids when we condense. And so when those sister chromatids split into daughter chromosomes, you're going to end up with the normal number of chromosomes at each pole. So you're going to have the diploid number of chromosomes at each pole. Anaphase is the shortest phase. So there it is. We can see a vague oval shape to this. And our sister chromatids here have split apart. Our centromeres have been cleaved. And so they're being yanked towards opposite poles. In telophase, the nuclear envelope reforms around the daughter chromosomes. The daughter chromosomes start to unwind, uncoil, relax, and decondense back into chromatin. So they become chromatin again. And because we've got chromatin reappearing, the nucleolus will reappear. So at each pole, nuclear envelopes reform. And then at each pole, those daughter chromosomes uncoil and unwind and relax back into chromatin. And as they do that, the nucleoli reappear. The nucleoluses reappear. So now they are daughter nuclei. The nucleoli reappear, and the chromosomes are unraveling into, well, I already said it, but just to be consistent, the chromosomes are unraveling into, say it a little louder. I can't hear you louder. You're gonna have to say it louder. Yeah, okay, I heard it that time. Yes, you're correct. Chromatin. So, as we get a regular old nucleus forming here, the mitotic spindle breaks down. So at each pole, we now have a normal nucleus and a single aster, which is what we started with in G1. So a nucleus, a nucleolus, and an aster is present. And each pole has in its own nucleus a full set of chromosomes, the diploid number of chromosomes. So all that's left now is to take our oval cell with two poles that have everything they need to be their own cell and pinch it and split it. So cytokinesis occurs. So at the end, telophase, the cells at the end of telophase look like G1. And the mitotic phase ends as cytokinesis occurs. So as it pinches inward, this inward pinch here at the center that I'm forming, right about here, this right here is called the cleavage furrow. And the cleavage furrow is just evidence that cytokinesis is occurring. And so it continues to pinch inward until, and it separates. And you get two genetically identical daughter cells. Cytokinesis, as I said earlier, is not a phase of mitosis. It just occurs at the end of telophase, and it's the formation of that cleavage furrow. Here we go. There we go. Now I've got a cleavage furrow. And then that pinches the cells, and because I was being ridiculous when I wrote this lecture, I decided to use twain again because I just like the word twain. It pinches the cells in two. In twain! And you have two complete identical cells. So here we are. This is the white stuff is the nuclear envelope reforming. These are becoming long and spindly because they're unraveling back into chromatin. And this right here is the nucleolus starting to reappear. Here's our single aster. And this is our cleavage furrow, this pinch along here. That's the indicator that cytokinesis is occurring. So, why don't I wait here while you guys put these in order from G2, prophase, prometaphase, metaphase, anaphase, and telophase. Tell me which is which. Okay, so let's start. Which one of these cells is a cell in G2 of interphase? G2 of interphase. Huh? What's that? Okay. Wait, no. You there with the Mac. Yes. What is your answer? No, actually, the answer is 5. 5 is the cell that is in G2. Okay, let's try this again. Let's go for prophase. Who's going to go for prophase? Give me prophase. You over there with the HP. Yes, correct. It is, in fact, 4. So, 4 is the cell in prophase. All right, how about prometaphase? We're coming along, narrowing it down. Now there's only 4 options, 25% chance of a random guess. Who wants to give me the answer? Who wants to give me the answer? Yes, you over there with facial hair. Correct. That's right. 6. Okay, so it goes G2 here at 5, prophase here at 4, prometaphase here at 6. Now we just need metaphase, anaphase, and telophase. This should be easy. So, how about you? Yes, you. Well, of course I'm talking to you. Who else would I be talking to? Which one is metaphase? Good. Yes, 1. All right. Last shot. 50-50 chance of getting it right. Give me anaphase. You. Okay. So you said 3. That was like a good guess. You're kind of a little lost, I guess. 3 is in fact telophase. No. 2. This one is the cell in anaphase. So this 3 makes telophase. So it goes G2, prophase, prometaphase, metaphase, anaphase, telophase. 5, 4, 6, 1, 2, 3. That is the correct ordering of these cells from G2 of interphase through prophase, prometaphase, metaphase, anaphase, and telophase. You will need to be able to draw these suckers for an exam. So you don't have to copy down these images, but just something that gets across the important things happening in each of these phases. So you'd have to draw a cell in G2 and tell me what's happening in G2. Like, what is the status of the cell in G2? So, binary fission is a term for what happens when prokaryotic cells divide. See, prokaryotic cells don't have a nucleus. So they also don't have the eukaryotic chromosome. They have circular DNA. So it's actually quite different when prokaryotic cells divide. They undergo a process called binary fission. For your purposes, until you take a microbiology class, you can think of binary fission as the mitosis equivalent in prokaryotes. In binary fission, prokaryotes duplicate their genomes and create two cells from one. So, binary fission is the prokaryote equivalent of mitosis. So while we're talking about mitosis, we might mention tumors. So, tumors. Tumors are when a cell divides uncontrollably. So naturally, cells have signals that tell them when to stop the cell cycle. Basically, the cell is sent a signal to start mitosis, and then it's sent signals to say, hey, you've divided enough, just hang out in G1 and do your thing. We don't need you dividing anymore. Sometimes it's as simple as density. Cells will continue dividing until they crowd in on each other, and then they stop dividing. Sometimes there's chemicals sent through your body that tell cells to start or stop dividing. Anyway, a tumor is when a cell divides uncontrollably. In other words, it no longer listens to the stop signals. So, if it's not listening to the stop signal, it's just going to continually undergo mitosis over and over and over and over again. And that's why a tumor is a mass of tissues. You have a single cell that goes rogue, and it divides into two cells that are identical, so they're both going rogue, and they become 4, 8, 16, 32, 64, 128, 256, 512, 1024, 2048, 4096, and so on. So, tumors can get out of control pretty quick. Now, there are two basic types of tumors. Both types are cells that have stopped listening to the stop signal. There are benign tumors. In a benign tumor, the cells are, of course, dividing uncontrollably. They're not stopping when they should be, but they stay in one place. The tumor doesn't spread. In a benign tumor, the tumor cells stay with the tumor. That makes a benign tumor relatively easy to be removed. I actually had two benign tumors, benign osteochondromas. They were tumors in the growth plates of my femur down by my knee. I had to get them cut out. They were benign tumors, so I was pretty lucky. I still carry a wicked scar to this day. I don't know how visible it is, though. Nah, it's not visible enough for me to want to shove my leg onto the table and waggle my scar in front of the webcam. Just imagine it's a totally awesome scar. Like, super impressive. I used to tell people I got into a knife fight with a drug dealer who was trying to push smack on kids. I won, of course, but not before taking a vicious wound to the knee. Anyway, we can contrast the benign tumor with the malignant tumor. In a malignant tumor, the tumor cells will shed. And when they shed, they enter circulation. And they'll move to other parts of the body. And wherever they land, a malignant tumor cell will start a new growth. It will start a new tumor. So as a malignant tumor increases in size, it increases the chances of shedding cells. And as it sheds cells, each cell lands somewhere else and becomes a new tumor. And so malignant tumors can rapidly spread throughout the body. They're released from the original growth, and they spread to other parts of the body and form tumors there that release cells, and those spread. So this release of tumor cells from the original growth is called metastasis. Metastasis is when a tumor cell is released from its original growth and moves to a different part of the body. So, malignant tumors are the big killers here. They can very quickly shut down organs because a benign tumor will grow on an organ and take up space, but it's predictable. You can get in there, cut it out, and you're good. Malignant tumor, it may grow multiple tumors on an organ. And as those tumors increase in size, they put pressure on the organ and they can start to take over the organ and sort of kill cells in the organ and shut you down. So, and even if you cut out almost all of the tumor, even just one tumor cell left behind could regrow the original tumor and give it chances to release. So malignant tumors, much more difficult to treat. Malignant tumors are cancer. So that took sort of a depressing turn. So here's a puppy. Oh, I'm Professor Dog, and I've designed a study in which I'm comparing bark to wolf. Oh, shut up. It's funny. Okay, last thing to mention before we move on to meiosis and gamete production is apoptosis. Apoptosis or apoptosis, depending on who you're talking to and just how they want to say it, is normal cell death or programmed cell death. Basically, this is when a cell is given a signal that tells it to terminate the cell cycle completely. Don't go through the normal processes of G1. Don't undergo mitosis. Just stop whatever you're doing and shut down. Apoptosis is completely natural. So it's not something to be like, oh, cells are being killed by apoptosis. It's more like, oh, good. Cells are being killed by apoptosis. That's good. So apoptosis is when some kind of signal ends the cell cycle completely and shuts down the cell, kills the cell. And then the cell dies and it breaks apart and its components are broken down and recycled and new cells use those nutrients. So that's the end of this video. We'll pick back up with sexual reproduction. So go jam a Q-tip in your ear to keep the knowledge from falling out.